For the Unit 3 review, we're going to go through the brain, the senses, as well as the autonomic nervous system. Part of the brain will also recall sensory and motor pathways. In the brain, we have 4 sections. In addition, there will be sensory and motor pathways. For part 1, you should know the location and function of each of the 3 cranial meninges. You should know that the pia mater is located on the surface of the brain. So any test question that refers to the surface of the brain, it's going to be about the pia mater, because the pia is the one that's actually adhering to the surface of the brain. The layer beyond that is the arachnoid mater, which has a lot of structures that's really similar to scaffolding, and that's because within the arachnoid mater you have cerebral spinal fluid that flows through it. So it provides more of a conduit for cerebral spinal fluid to flow through. Within the brain, we're going to learn about cerebral spinal fluid, where it's formed, but where it comes through the arachnoid mater is actually on the outside of the brain, the outside of the spinal cord, and it's coming back up. So when it's coming through the arachnoid mater, it's actually coming up the spinal cord and then around the outside of the brain to then collect at the arachnoid villi, which are the granulations that actually return cerebral spinal fluid to the blood. And then the third cranial meninge is the dura mater. The dura mater is the tough, strong outer covering. It's really similar to a paper bag, a wet paper grocery bag. Has that feel, that thickness. So if you were to, the dura mater is fairly substantial, but it's also very, very strong. The formation and reabsorption of cerebral spinal fluid, we know that cerebral spinal fluid is created within the ventricles of the brain. The structure that makes cerebral spinal fluid is the choroid plexus. The choroid plexus is granulated tissue at the floor of each of the ventricles within the brain. It is at this cerebral spinal fluid is formed, and what is in cerebral spinal fluid is really clear fluid. There's no blood in it. It has glucose, it has oxygen, it just does not have very large molecules. So you're not going to get a lot of the circulating hormones through the body is not going to end up in the cerebral spinal fluid. Some do and some don't. If it's a lipid soluble hormone, that would be any of our sex hormones or our steroid hormones, they would indeed be able to cross because of their lipid-soluble nature. However, the cerebral spinal fluid does not include red blood cells, does not include most large protein molecules in the blood. So cerebral spinal fluid is made by the choroid plexus. It then travels through the ventricles within the brain, and then it goes down the central canal, back up the outside of the spinal cord through the arachnoid meninges, around to the top through the arachnoid meninge to the arachnoid villi located at the top part of the brain, and it interfaces between the arachnoid villi, returning the cerebrospinal fluid to the blood. The blood is actually within these 2 layers of dura mater. We didn't talk about the dura mater actually being 2 layers, but technically it is, and the cerebrospinal fluid enters that as it goes into blood. So the blood's within the dura mater actually then helps to hold and form blood vessels that's exiting the brain. You should know the pathway of cerebral spinal fluid. You should know that that the lateral ventricles 1 and 2, they're sort of counted as 1 and 2, but they're not numbered. So they're both lateral ventricles. They are in either cerebrum. They arch around the diencephalon. The fluid from there drops down into the 3rd ventricle, which is located between the 2 thalami. Then the fluid goes from the 3rd ventricle to the 4th ventricle through the cerebral aqueduct, also known as the mesencephalic aqueduct, but cerebral aqueduct's a little easier to remember. So know that the cerebral aqueduct connects the 3rd ventricle to the 4th ventricle. The fourth ventricle is a little triangular shaped ventricle that's located anterior to the cerebellum and posterior to the pons. Once fluid has left the fourth ventricle, there is a little channel that brings the fluid straight into the central canal of the spinal cord, where it then drops down through the very center core of the spinal cord, only to be released at the bottom around the cauda equina, and then it comes back up to the top of from the outside of the spinal cord to the top of the brain via the arachnoid mater. The composition of cerebral spinal fluid is watery. It contains oxygen, water, PAF. It has a moderate pH. It regulates many things. There are various neurotransmitters within there. There are hormones that are in there. But what's important is what we block out. We block out large molecules, most of the circulating hormones throughout the whole body. And it allows for lipid-soluble substances to cross the cerebral spinal fluid. And that is through the blood-brain barrier. Within the blood-brain barrier, it is the separation between the blood and then what we allow for the actual neural tissue to be exposed to. So the blood-brain barrier is very effective at keeping a lot of things out, which is why it's very, very difficult often to create pharmaceutical drugs that's actually going to affect specific parts of the brain, because the blood-brain barrier is so effective at keeping a lot of these larger molecules out, which many of the pharmaceutical drugs are. The regions of the brain are, first, the cerebrum. That's what we're going to talk about in this section. The cerebrum is the recognizable part of the brain. It's the wormy component of the brain. It has the outer cerebrum. We have the cerebellum, which is known as the tiny brain or the small brain in the back. We have the diencephalon, which is inside. And then there is the midbrain and then the brainstem. So those are the main areas of the brain. The region within the brain of white matter that's the most recognizable. There's lots of white matter regions, but the one that's most recognizable is the corpus callosum. This is connecting the right and left sides of the brain. It is filled with axons that transmit information side to side within the brain. The divisions of the cerebrum specifically are the lobes. We have the frontal lobe just behind the forehead, and we have the occipital lobe way on the back side of the brain. We also have the two parietal lobes, which are located on the posterior superior part of the brain. So it's right behind the frontal lobe, but right above the occipital lobe. And then we have two temporal lobes on either side, just underneath the ear. The motor areas and higher order thinkings are within the frontal lobe. So when we talk about motor areas, we have a division within the brain known as the central sulcus. First, you should know the central sulcus extends from the longitudinal fissure. The longitudinal fissure is the big crack between the right and left sides of the brain. So if you have a brain sitting on a tray in front of you, there is a large crack right down from front to back that divides the brain into the right and left hemispheres. From that longitudinal fissure is a specific sulcus that extends out and down. That sulcus divides the frontal lobe and the parietal lobe. In front of that sulcus, which is known as the central sulcus, you have the precentral gyrus. So a gyrus is the mound portion, whereas the sulcus is the crack between the mounds. So in front of the central sulcus, which divides the frontal and parietal lobes, you have the precentral sulcus. The precentral sulcus is also known as the primary motor cortex. And the post-central sulcus, the one right behind, that's part of the parietal lobe, is known as the primary somatosensory cortex. We'll go to the premotor cortex side. The premotor cortex within the frontal lobe is the part of the brain that I like to think of as the strings of the puppet. We have a premotor cortex that I like to think of as the puppet master himself. So if premotor cortex is the area of the brain that's going to plan a movement, whether it's a musical piece, someone's playing a trumpet, or someone's kicking a soccer ball, or someone's doing archery and pulling an arrow on a bow and releasing it. The sequence of muscles required to hold the bow, grab the arrow, pull it back, or the sequence and the amount of force required, say, to play the piano, play a specific note section or a specific music section on the piano, that is planned out. That order and how forceful or how light is planned out by the premotor cortex. It then tells the primary motor cortex, which is also known as the precentral gyrus. That is the area that's directly connected to all of the skeletal muscles within your body. So the premotor cortex selects which muscles on the primary cortex are going to get recruited for a given sequence. So if you're practicing a sport or practicing any type of movement, it is your premotor cortex that you are fine-tuning the recruitment pattern in. Now, also another motor area there is known as the Broca's area. That is also muscles, but muscles that are specific to forming words. And this is only on the left side. So the motor area for muscles that's going to form words of your mouth, whether it's your throat and the formation of your mouth and your tongue, that's going to create speech. And this is from Broca's area, and it's only on the left side of the brain. Whereas the right side of the brain in that same area actually puts emotion into the words that you're saying, whether someone's very excited and they're putting that emotion in the words they're saying, or if someone's being sarcastic and they're saying what the words are saying, but the emotion is actually giving the opposite impression to the meaning. So the left side area of Broca's forms words. The right side puts emotion into the words. The higher order thinking in this category means future planning, goal setting, personality. It's your almost restraint. If you're sitting at a buffet full of sweets and some kids may just have no restraint and you want to just eat their fill, versus someone having a sense of restraint and propriety and maybe just taking one of them. So it is this higher order thinking that you're controlling yourself in a way, but you're also considering future implications in this, as well as understanding interpersonal relationships. So that's the higher order thinking. Somatosensory areas of the parietal lobe. We have the post-central gyrus, that is also known as the primary somatosensory cortex. The post-central gyrus is where we're receiving all kinds of sensory information from the body. It comes up through the spinal cord, crosses over at the medulla oblongata, goes up into the thalamus, and then out to this primary somatosensory cortex, where it is giving us a feeling, whether it's a pain or temperature, any of those mechanoreceptors that we're going to talk about in the sensory motor section. That information is coming up to this primary somatosensory cortex. Right adjacent to it in the parietal lobe is the somatosensory association area. Now, an association area is where we're interpreting it. So as this information comes out to the primary somatosensory cortex to tell us the part of our body, the type of sensations that part of the body is feeling, then it's that somatosensory association area that's going to figure out what is going on. Are your hands in a bucket full of rocks and they're feeling sharp objects, but you know that they're rocks? Or are you running your hands down a blanket and you can feel soft fabric? You based on life experiences, you're able to determine what your sensations are telling you about the world around you. The temporal lobe has functional regions being the primary auditory cortex. So from your inner ear, we have information from the cochlea, which gives information regarding hearing. So you're going to get volume and pitch. That's coming to the primary auditory cortex, and then around that is the auditory association area that's going to interpret the sounds, whether it's a baby crying or tires squealing. You also have this in part of the temporal lobe, but it also extends through the parietal and occipital lobes, is going to be Wernicke's area. And so the Wernicke's area in this context helps you, because it's part of the hearing component, helps you understand someone else's words. So it is receiving someone's words. So you can call understanding language. You're able to interpret the words somebody else is saying. Now, the Wernicke's is only on the left side. The right side is going to be understanding the emotion in someone else's words. So this is part of the temporal lobe, but it also it blends out into the parietal and occipital depending on person to person. So it's less defined, it's pretty well defined, but it sort of transcends some of these lobes. Also in the temporal lobe, on the medial inside part, is going to be the region for smell. Our smell sensations are located within there, and they are also adjacent to our memory areas. So you should be well aware that the memory and smell are actually associated together. At this point, you should just know smell is located on the interior portion of the temporal lobe. Taste is located off of the primary somatosensory cortex in the parietal lobe, and vision is going to go to the occipital lobe. That's going to be in the back of the brain. So we have the primary visual cortex. The primary visual cortex is information from the eyes about light intensity, about color, about shading. It gives all the information from rods and cones. And we actually get very specific sections of our eye are going to different parts of the brain. It is around this area, the primary visual cortex, that we have the visual association area. So the visual association area is where we interpret what we see. You're seeing kind of a white blob. Is it a ball or is it a flower? So your life experiences are going to tell you what you are seeing. The location functions of the language integrative areas. So we've talked about Broca's area forming words and Wernicke's receiving or understanding somebody else's words. That's the left side. The language integrative areas on the right for the Broca's is putting emotion into the words you're saying, whereas the language integrative area or language affective area on the right side for Wernicke's is understanding someone else's emotions. So if somebody is saying, I'm doing just fine, but they're saying it while they're crying and their voice is quivering, then the language integrative area for Wernicke's, the understanding of emotion says, although the words say they're fine, their emotions are telling me that maybe they are not fine. So that's your awareness of somebody else's feeling and their position in life. And then there are the electroencephalogram waves, also known as EEG waves. So we're going to have alpha, beta, delta, gamma. So you want to think of alpha as just being mellow, really just mellow and at peace. Beta, you want to think of focused and concentrating. And so if you're taking a test right now or going to be taking a test, you're going to have beta waves. And then you have delta and gamma. They are a bit of a mix and a little harder to interpret. So we'll sort of we'll just talk about more gamma, more delta waves. And so both of them are going to be more involved in sleep, or they vary depending on if it's children or an adult that's got some sort of mental dysfunction. But in essence, you want to think of, just for simplicity for our exam, you want to think of them more associated with deep sleep and more subconscious type patterns. So think of more, let's just think on the positive side and less on the pathology, more of various stages of sleep, and just think of sleep, deep sleep. For the diencephalon, we have the thalamus, which is the heart and soul of the diencephalon. We have the epithalamus, which is above it. So the thalamus are two egg-shaped structures within the brain that the cerebrum wraps around. We have the epithalamus that comes above it, and the epithalamus will actually terminate in the posterior side in a bulbous point that's known as the pineal gland. In the pineal gland of the epithalamus, that's where, in the absence of light, it's going to release melatonin. The thalamus is the main area of our brain that's more like a switchboard. It is helping to direct mostly sensory information, but it can direct a few others, but it's really helping to send information from one part of the brain off into the appropriate processing region of another part of the brain. Then we have the hypothalamus. The hypothalamus is below the thalamus, and it is a monitoring center for your whole entire body. The hypothalamus monitors whether you're thirsty or hungry or hot or cold. The hypothalamus has a lot of input throughout your body, and it is going to be the master controller of your pituitary gland, which is then going to send out a number of hormones throughout your body to elicit changes based on whatever the hypothalamus detects is out of whack. The role of the hypothalamus and the relationship of the hypothalamus and the pituitary gland is going to be a main topic that we'll start off Bio 202 with when we do the endocrine system. Then the specific features of the hypothalamus is we would have the infundibulum, which then connects it directly with the pituitary gland. We also have within this region the mammillary bodies, which are associated with smell, and I think in the notes we have smell and some emotions, which is part of our limbic system. But newer research really puts mammillary bodies also as a main area with regard to memory, and they're just trying to figure out its components. But some patients that are missing mammillary bodies have a series of amnesic episodes, and so there's a role there. So for the purpose of our class now, you should understand that the limbic system is really combining your emotions, smell, and memory. So as the example of, walking into a house that you can smell cinnamon rolls, that's going to make you think of, lovely thoughts of your grandma and make you happy. So those are all things, or a love letter that with perfume on it. If you spray perfume in a love letter, you're hoping to evoke somebody's limbic system, whoever you're sending it to, so that they will smell it, think of you, remember you fondly, and have wonderful memories of your experience together. So that's what the limbic system is. With regard to memory, the specific parts of the memory are the hippocampus and the amygdala. Those are the main areas for memory, in addition to mammillary bodies, as well as a number of other regions. The main one I want you to focus on for memory is the hippocampus. It is also known that the hippocampus is the area of the brain specifically that is being degraded in people that have Alzheimer's disease. The mesencephalon is right between the diencephalon and the brainstem, so it's known as the midbrain. The midbrain has a number of features. First is the cerebral peduncles. The cerebral peduncles are two beams of neuron tracks that actually separate to go to the right and left sides of the brain. So as neuron tracks are coming up through the brainstem, they actually branch out to go to the right and left hemispheres, and that is going through the cerebral peduncles located in the midbrain. On the posterior surface of the midbrain are another important structure known as the corpora quadrigemina. There are 4 bumps on the posterior side of the brain underneath the cerebrum but above the cerebellum. You will have superior colliculi and inferior colliculi in this corpora quadrigemina. Quad tells you it's 4. So there are 2 superior colliculi, which allows your eyes, so vision tracking, so it connects with your muscles of your eye as well as your vision, so that you can move your eye to match what you're seeing. Like if you're going to watch a tennis ball go back and forth, it's actually your superior colliculi that's helping you track that ball. The inferior colliculi are hearing, so it's actually moving your head so that your eyes can focus on the origination of a particular sound. So the inferior colliculi are helping you triangulate between your ears where a sound is coming from so that your head can appropriately move in that direction. The cerebellum is known as the little brain. Inside the cerebellum is white matter known as arbor vitae. The role of the cerebellum in balance and equilibrium is that it provides proprioceptive input. So when we talk about proprioceptors, they're sensors that are located out in our joints. So they tell us where our body is in space, whether our arms are above our head or out to the side. We don't have to see to know where our body is in that position. And so that's located in the cerebellum. And therefore the cerebellum is an ideal location for when you have a motor movement. Your premotor cortex in the frontal lobe tells your primary motor cortex which muscles to move. But based on your sensory feedback of what your body's experiencing, are you, just standing up still on a flat surface and you just have to recruit muscles to help you stand up? Or maybe are you standing in water where there's waves moving and now you have to counterbalance the motion of the waves? The cerebellum helps provide your body with feedback so that you can make the appropriate muscular adjustments. And then I skipped the cardiovascular and respiratory centers. So the parts of the brain stem, so we're focused on the second item here. The parts of the brain stem that you need to know are the pons and medulla. The pons helps with breathing and the depth of breathing, where the medulla oblongata is the part that's more the rate, but the medulla oblongata also helps control the cardiovascular system. So the medulla oblongata has both a respiratory and a cardiovascular component to it, where as far as we know, just the pons are all, as far as we're going to need to know in this class, is focus on just the pons being the driver of the respiratory system. Within this area, the brainstem, midbrain, I want you to also be aware of our reticular activating system. It is not a specific part of the brain. It is located within this area, but it is actually a functional system that helps us be aware or tune out things. So it is to do with our consciousness. Our consciousness more about how much we're aware of what's around us. So if you're in a room and say, there's a ticking sound, perhaps it's a clock. That's, to some people, it would be extremely annoying because their reticular activating system is having you hear this noise, the tick, tick of a clock. Whereas other people, their reticular activating system may be filtering it out a little bit more and they don't even notice it at all. So that's just one subtle example. If someone's in a coma, it is their reticular activating system that's actually shutting down this input to their conscious brain, so that if you're shaking someone's hand and trying to wake them up, that information is not actually making it up into the brain. So they're not aware of you shaking their hand because of that consciousness level. And that's also located in this region of the brain. So sensory and motor processing. We have sensory receptors, so you should know 5 main types of receptors They would be the nociceptors and the baroreceptors and the mechanoreceptors and the chemoreceptors. So there's these different types, whether it's about pain or pressure. Then you should know the classes of mechanoreceptors. Those are the types that are touch and pressure. You might have deep pressure or light pressure. We're less focused on these as we were when we did the skin and those type of sensations. But I want you to know that they are in the mechanoreceptor categories and a sense of what they do. Proprioceptors and baroreceptors. The baroreceptors at the bottom are really about pressure, and those are found more in arteries, so determining whether someone's pressure is very high or low. We have baroreceptors in other areas, but the arterial ones are probably the ones I'd like you to focus on. Proprioceptors we talked about already with regard to the cerebellum. They are about where your body is in space, and it has much more to do with you being aware of what's going on in your body, not just by touch, but by position. Then sensory processing. All these receptors come together to really bring information up into the brain. It comes up into the brain via the brain stem, crosses over at the medulla oblongata. That means pain on the right side of your body is actually going to get processed on the left side of your brain. And it's going to go up to that primary somatosensory cortex, also known as the postcentral gyrus. It is in the parietal lobe. It is this primary somatosensory cortex that brings in information about pressure or pain or twisting pressure. All the different tactile receptors and other receptors are actually terminating in this part of your brain that then the somatosensory association area will interpret. Referred pain is pain from your organs. We do not have our organs mapped out very well in our brain. So when there is some sort of damage to an organ or one of your autonomic, meaning that you can't control parts of your body, that pain actually follows, it piggybacks on other sensory pathways. So someone may have pain, say, in their heart if they're having a heart attack, but that stress and pain is coming into the brain, piggybacking on the same sensory pathways as would be the skin of your arm on the left side. So that's why heart attacks can be misinterpreted as just a pain in someone's arm. And so now that's a little more, it's more common to know that, but that's just one example. Other areas of your organs will also have associated referred pain, and it's a bit of a ghosting type pain because it's not always, you might feel in your brain as thinking you have specific pain in an area like your shoulder, but really it has to do, what's really undergoing damage is something maybe in your abdominal cavity. So that's why it's referred. It's really not pinpointing exactly where it is. Motor control, we talked about the frontal lobe, where that premotor cortex plans out a sequence of movements. It tells the primary cortex exactly what muscle to contract and how much and when to end the contraction. So that is causing us to move. The cerebellum gives it feedback regarding your balance and equilibrium and proprioception to fine-tune that. For the special senses, we have 3 main parts that we went through. There are 5 special senses: olfaction, which is smell; gustation, which is taste; vision, obviously is sight; equilibrium is balance; and hearing. For olfaction and gustation, you should know that both smell and taste require the molecule to be dissolved. The airborne molecule gets dissolved in your nasal cavity through the mucus in your nasal cavity, and that is how it gets dissolved to be able to adhere to your olfactory receptors and then go into the brain. The gustation is the food that's in your mouth has to be dissolved by saliva, and that's how your taste buds can pick it up. So you should know there are a number of different taste and smell sensations. I don't believe I have specific test questions on what these sensations are, just because they vary. Every time you read a research article about it, there's some variation to it. But just know there's a myriad of taste sensations. We actually have many more smell sensations than we do taste. which is why most of the food that's produced nowadays have a greater smell component than taste, which is why when you're sick and you have a lot of extra mucus in your nose, that we actually don't dissolve the smell. It's too far for the smell to dissolve, that it doesn't make it to our olfactory receptors. But if you can't smell the food, it doesn't taste very good. And that's because the majority of our pleasure from eating food that we interpret as taste is in fact the combination of taste and smell. So the neural pathways is that for smell, we don't go through the thalamus. For the smell, we go from the olfactory nerves dangling in the nasal cavity. It targets the olfactory bulb and then the olfactory tracts coming into the brain to the hypothalamus and then to the temporal lobe, and it's part of the limbic system as well as other parts of the brain. But the limbic system, I wanted to point out, is the part of the brain that connects our emotions with our memory to smell. For taste, we have 3 cranial nerves that are involved in our taste sensation. It then goes into the brain via the medulla, but I want you to notice that it goes to the thalamus. So smell did not go through the thalamus, but the thalamus is where we're going to direct it up to that primary somatosensory cortex region. For vision, you should know the accessory structures of the eye. That means your palpebra, which is your eyelids. We have the conjunctiva, which is our blood vessels that are serving the outside of the eye and goes to the inside of our eyelids. We have the muscles that are moving our eye around. You should know the rectus muscles are straight. So if it's a superior rectus muscle, it's going to actually lift your eye up. Inferior rectus moves your eye down. Know the oblique muscles mean they're coming at an angle. So the superior oblique muscle is actually going to turn your eye down and out. So the oblique muscle's action does a bit of the opposite to what the name is. And then we have lacrimal glands, which are located superior and lateral to the eye. That's what forms tears. With regard to the tunics, we have the outer layer of the eye. Tunic just means layer. So the outer layer of the eye is the sclera, the white. That is the white of your eye. And then the sclera on the very anterior portion, the part that maybe someone would put a contact lens on, that's the cornea. Just under that, we have another layer that's known as the vascular tunic. And in this layer, we have the choroid in the back, which is black. It actually absorbs and minimizes light. And then it comes anterior, but on the back side of the lens, to be the ciliary body. The ciliary body has 2 jobs. The first job is to actually contract, and it will actually move the lens, contract and relax. So it actually can allow you to focus close up or far away. The other job of the ciliary body is to make aqueous humor that's going to now fill up the anterior part of the eye. We also have the neural tunic, which is the innermost layer of the eye, and that is the retina. That's actually the nerve component that we're going to see with. The internal chambers of the eye, you really want to think of the lens as dividing the front part of the eye from the back part of the eye. So the posterior chamber of the eye contains this very jelly-like substance, clear, but it stays in your eye for your whole entire life, and that's known as vitreous humor. The anterior chamber of the eye between the lens and the cornea is a smaller space, but it's filled with aqueous humor. It's more of a watery, but you're constantly making new aqueous humor and draining it out via the canal of Schlemm. So in the anterior chamber of the eye, the ciliary body makes aqueous humor. It bathes the front of the eye. It provides nourishments and nutrients to the cornea as well as the iris, and then it drains out via the canal of Schlemm. The retina components, which in the posterior part of the eye has rods, cones, and then there are the other nerve connections, bipolar cells, ganglion cells, and then the axons, Well, from all of these rods and cones, the rods and cones are the ends of the nerve where they're either detecting black and white in contrast, that's rods, or color, that are the cones. So the axons of every single one of them throughout the entire retina on the back of the eye come out through an area known as the optic disc. Within hearing and equilibrium, we should know that our external ear is known as the auricle or pinna. We go through the external auditory canal into meet the tympanic membrane. The tympanic membrane is your eardrum. Then inside that is the middle ear. You have 3 ossicles, the malleus, incus, and stapes. The 3 of them move together like gears that actually amplify the sound waves from the tympanic membrane. And it pushes onto the oval window, which then enters the cochlea, sending these, it's like a plunger, moving, like making a wave pool of movement within the cochlea. Now each wave, whether the wave is fast or the wave is slow, is the frequency. That tells you the pitch or what sound you're listening to. The magnitude or the size of the wave is going to tell you how loud it is. The vestibule in the inner ear is the area that we actually have our static equilibrium. That's if we're leaning over or looking down a canyon side, but we are still, but we're just leaning to a position. Whereas the semicircular canals are more of our dynamic equilibrium. So within the vestibule, we have these little features known as the utricle and the saccule. Those are the ones that are telling us whether we're accelerating, like if you're sitting in a car, you physically are still, except for the car is now moving. So it's giving you a sense of motion. That's the utricle and saccule in the vestibule. Now the semicircular canals, that's more if you're spinning, you're going to do a somersault or a cartwheel, or you're spinning like a top on a, doing some sort of pirouette. Those are semicircular canals and they're moving, and that's your dynamic equilibrium. And the sensory receptor there are known as the ampulla. The last thing, but it's not listed here, is what we hear from in the inner ear, which is the cochlea itself. The cochlea is getting those sound waves from the ossicles. They're moving, they're bending hair cells. It is the location of the hair cells that get bent based on the frequency of the sound waves that's going to actually tell your brain what pitch that you're hearing. For the autonomic nervous system, we have these 4 parts that we're going to go through. The 2-step pathway and the basic overview, really just for the overview, I want you to know about the parasympathetic and sympathetic nervous systems in general, and then we'll go through them a little more technically in the following conversation. So the parasympathetic is more as you're just mellow. Our bodies are in the parasympathetic system the vast majority of our time. So it is not equally divided between the parasympathetic and sympathetic in terms of our use of these systems. We are primarily functioning in a parasympathetic role. When we're stressed out or we're excited or we're exercising vigorously, then we move into the sympathetic nervous system role for a little bit of time, but we're mostly in a parasympathetic mode. We also know that both of these systems utilize a 2-step pathway, which is different than controlling just your skeletal muscles. Your skeletal muscles have just one nerve coming off the spinal cord and going straight to that muscle. So that's just one step. Both the parasympathetic and sympathetic nervous systems utilize a nerve that comes off of the spinal cord, but that nerve doesn't take you all the way to the heart or the gland that you're actually trying to activate. It stops and you have to synapse onto a second nerve that will then take the impulse the rest of the way to whatever autonomic, whether it's gut action or whether it's your heart or glands or. So what you should know from the 2-step pathway is that your preganglionic neuron will go off of the spinal cord. It will then, the ganglia is the location that it's going to synapse onto a postganglionic neuron that's going to be taking it all the way to the actual organ. The sympathetic nervous system, within there, it's important that you know that it's called thoracolumbar, meaning it's only using the spinal nerves from the thoracic and the lumbar region. Those thoracic and lumbar spinal nerves come out, and where the synapse is going to take place, or the two-step pathway, is a whole series of chain ganglia that actually they're more chain ganglia than the specific spinal nerves coming off of your spinal cord. So these chain ganglia extend out on either side of the vertebral column. They're also known as paravertebral ganglia. So these spinal nerves go out to the chain ganglia where a synapse will take place so that your second neuron is then going to leave the chain ganglia going out to the organ or tissue that you're going to be stimulating through the sympathetic nervous system. The sympathetic nervous system has also collateral ganglia, but that's exclusive to the abdominal cavity, because within our abdominal pelvic region, pretty much everything in there is autonomically controlled. We're not telling our ovaries to ovulate, or we're not telling our large intestine to move food along. We're not telling our small intestine to inspect, small food along, and telling our pancreas to release digestive enzymes. So that is under an immense amount of autonomic control. So collateral ganglia are a second ganglia point that any of the spinal nerves that have the first nerve coming off, it actually goes right through the chain ganglia to collateral ganglia, and that's where the synapse is going to take place. So the second nerve can come off from the collateral ganglia to various regions of your abdominal cavity. And so the ganglia are really specific. You have the upper, middle, and lower part is really how they're divided. And finally, for the sympathetic nervous system, we have the adrenal medulla. It's part of our adrenal gland, the central part. And what it does is we have one nerve coming all the way from the spinal cord, all the way to the adrenal medulla, no synapses, no ganglia, straight from the spinal cord to the adrenal medulla. And then instead of synapsing onto a second neuron, it just dumps out what that neuron would have released as a neurotransmitter. It dumps out norepinephrine. So it doesn't need a second neuron, but that way we get norepinephrine dumped into our bloodstream, circulating through our body. So norepinephrine is important, although I'm getting ahead of myself with regard to neurotransmitters. Norepinephrine is important because that is what ultimately the sympathetic nervous system releases to the heart or the glands or various regions of our abdominal cavity that's going to cause it to respond to your flight or fight or sympathetic system. So if norepinephrine is released and it touches and it goes to the heart, whether it's through the chain ganglia and a second neuron, or whether it's through the blood from the adrenal medulla, your heart rate's going to increase. The parasympathetic nervous system is known as craniosacral. It uses cranial nerves, and you should know which ones, as well as sacral nerves. It also utilizes a two-step pathway. So even though these are the only nerves that are coming off of the central nervous system, the brainstem and spinal cord, it still can affect regions all over the brain, all over the body. That is because the vagus nerve, which is known as the wanderer, goes to so many of our autonomic nerve tissues. So the parasympathetic nervous system works because at the level of the tissues, if we use the heart again as an example, it will release acetylcholine. So if the heart sees acetylcholine, or acetylcholine touches the heart, and it's only going to touch the heart through a second neuron on this parasympathetic nervous system, then the heart rate will slow down. So the bottom line between the parasympathetic and sympathetic is parasympathetic, we are in that state much more than we are sympathetic. Our organs respond in a parasympathetic way, mellow meaning low heart rate or increasing digestion in a way because acetylcholine. The sympathetic nervous system will reduce digestion and raise heart rate in a flight or flight manner because it's going to release and expose the heart to norepinephrine. So think of acetylcholine and norepinephrine as the on-off switch, or the switch that determines whether you're parasympathetically stimulated or sympathetically stimulated. So let's get more into detail with the neurotransmitters. You should know that in this two-step pathway, no matter if it's parasympathetic or sympathetic, that first neuron will always release acetylcholine. In the ganglia, the dendrites of the second neuron will have a nicotinic receptor. A nicotinic receptor is going to receive acetylcholine at the ganglia. In the parasympathetic world, the ganglia is right there at the level of the organ, so the second neuron is very, very short. In the sympathetic, we know the ganglia are really close to the spinal cord. The chain ganglia are, even the collateral aren't that far away. And so it is the second neuron in the sympathetic that's very long. So when with the second neuron, both of them, whether it's parasympathetic or sympathetic, will have nicotinic receptors that's going to be activated by acetylcholine. Then, if it's parasympathetic, it will go out to the tissue or organ, and if it's parasympathetic, it will release acetylcholine onto a muscarinic. So this is a different acetylcholine receptor, a muscarinic receptor. Whereas in the sympathetic nervous system, that second nerve will release norepinephrine to either an alpha or beta receptor. Dual innervation means that an organ, and we'll keep using the heart as an example, is got both innervation. It has both parasympathetic and sympathetic influences. The autonomic reflexes, examples of those would be your mouth is watering because you smell good food, or your stomach is growling because you're hungry. Those type of things you have no control on, but it's your autonomic nervous system that's actually creating some effects. So the main thing you should know is there's a two-step pathway. You should know the first step, what neurotransmitter is released, and that would be acetylcholine. You should know for the second step on that neuron, on the dendrites of those neurons, you should know that there are nicotinic receptors for both parasympathetic and sympathetic. You should know the parasympathetic has a very long preganglionic neuron and a short postganglionic neuron. You should know the sympathetic has a very short preganglionic neuron and a long postganglionic neuron. And then you should know that the second neuron, whether it's parasympathetic or sympathetic, those are the ones that are going to be different from each other. Sympathetic will release norepinephrine to be received by an alpha or beta receptor on the tissue or organ, where parasympathetic will release acetylcholine to be received by a muscarinic receptor.